me share my screen. Okay, so hi everyone. So let me get started with the introduction of my lab, ATCG lab. So we call it ATCG. It stands for Algorithmic Techniques for Computational Genomics. We work at the intersection of computer science and biology. So I'll give more details in the next few slides. The lab URL of our is at-cg.github.io. Again, you can find all the information like some of the projects, students, publications, software, all the information is available on the lab website. Okay. Yeah, so I think very similar to what Professor Deepak just gave you the introduction, right? So similar to Quest Lab, we also hire students from diverse backgrounds. So for example, we have 2 PhD students right now. Our lab started in 2020. So we have Ghansham, who is a second year PhD student, and Sudarna, who is a first year PhD student, right? There are 4 MTech students with us, 1 UG student, and 2 project interns who work with us remotely from their home institutions. So in terms of the background, again, as we saw in the previous lab also, so students do come from diverse backgrounds into our lab also, but of course they have a common passion of working in this area, as well as fundamental computer science knowledge of algorithms, as well as programming. Okay, so what we do. So as you may be knowing, so every living cell has DNA in it that codes for its function and structure. So for example, in human cells, we have a human genome. With each cell has human genome, which has about 3 billion DNA characters in it. So if you open a cell, inside you will find chromosomes, and each chromosome can be seen as a thread of DNA. And DNA can be interpreted as a sequence of nucleotide characters, A, T, G, C, and so on. So what we are doing in our lab is developing algorithms that help interpret these DNA molecules. Now, why do we need algorithms to interpret DNA, right? So what happens in any hospital or biological lab which is interested in genome sequencing is first step, they will take a clinical sample. For example, it could be a DNA sample of a patient, right? So DNA in its natural form, for example, if I just spit out some saliva, it won't, you cannot recognize my DNA from just the saliva, right? So we need some sequencing mechanism to extract the DNA from the sample and some instrument to be able to read the DNA from the sample. So we have various type of sequencing technologies available today that can help sequence the DNA in a sample. These sequencing instruments are capable of producing 1 terabase data, so 1 terabyte data per day. And that's where you can imagine that if these instruments are producing more than 1 terabyte data per day, one needs clever algorithms to help analyze this data. Right? So this data is really massive, and to a life scientist or a doctor who is interested in drawing conclusions from this data will need efficient algorithms to make any conclusions, right? So one thing I want to mention at this point is that these sequencing instruments do not read the genome from one end to other. What they will give you is small fragments of the genome, because due to technological limitations, we cannot read a genome string from end to end. We can only parse small fragments of the genome at a time. So if you put the genome sequence into the instrument, you will get small pieces of the DNA as output, right? So So using those small fragments, one has to perform some computational analysis depending on what application needs to be addressed. And that application will be used for various health and biomedical applications. Right? So for example, if it's a patient, they may want to understand what mutations are there in a patient and what are the disease that they are more likely to develop, or what are the underlying causes of a disease that they may be already be suffering from. Right? So there are many applications actually in various sectors. So for example, you can consider agriculture, infectious disease, cancer analysis, evolution, even forensics, and birth care health. So at the time of birth, both mother and kids are typically screened, their genomes are typically screened to analyze any risks involved. So just one example, right? So in agriculture, why would you do genome analysis? One would do genome analysis to improve the crops going forward. For example, you may want to develop crops that are more that can adapt to heat going forward because of global warming. So one needs to analyze what genes what genes enable a plant to adapt more to more heat, right? So these type of questions are always analyzed using genome or RNA analysis. And these are the application sectors where our software get used eventually that we develop in our lab. So one, as I mentioned already, that this cost of, so the genomic data is very big. And that has happened over the last few years because the cost of sequencing has dropped by orders of magnitude. So if we look at few years back, there used to be maybe 1 or 2 sequencing centers around the world. But now every life science lab, like even if you visit IISc, a lot of labs now have the ability to sequence genomes because the cost of sequencing and the ease of sequencing has become so much better compared to past 15 or 20 years. So as a result, the amount of data that is available in public databases has grown exponentially. Just to give one other example, so it is estimated that we will have 60 million human genomes sequenced by 2025. So various countries around the globe have their national sequencing initiatives going on. In India, we have a Genome India initiative going on, which is doing whole genome sequencing of 20,000 human genomes already. So as a result of all these projects, genomics is actually estimated to become the biggest source of data on the planet, overtaking any social media or any other domain. So given this, naturally you need scalable software and big data software to be able to interpret this data and draw meaningful conclusions, right? So in the next few slides, I will give some examples of the projects that we are doing in the lab. And if you are, so I'll give a very high level description so that it is, you can understand it. But for more details, you can always visit our lab website and check out the publications. Okay, so yeah, so basically the idea that motivates us in our lab is we want to empower biomedical scientists with efficient algorithms, right? So we want to give them software that they can use to analyze the data that they generate. Now, when we develop algorithms, we are also interested in mathematical formulations and provable guarantees. So we don't want to just give a software which we think will work on some of the data sets that they have. We also are interested in giving mathematical guarantees, okay, to better analyze when the algorithms will work and when the algorithms will not work. So that is done using various proof techniques, right? And then ultimately, based on the algorithms that we develop, we want to develop prototype software that we can open source and give them on GitHub for the community to use. So on the computer, so there are various sequencing technologies available with different characteristics. There are many applications in this domain that we want to address. On the computing side, we want to employ different categories of techniques. So for example, we want to develop new graph algorithms, string algorithms, sketching techniques, parallel computing techniques, integer linear programming. So these are some of the standard computer science techniques which we want to adapt to the domain and address some of the domain specific challenges. Right? Okay. So let me move on to some of the project examples that we are working in our lab. And based on that, you will have an idea of what are the challenges involved. So first project is I refer to as genome assembly. So this is so genome assembly is the task of reconstructing the genome. So as I said, when we have a complete genome sample from an individual, the genome in its natural format is not accessible to us. We need sequencing instruments to be able to read those parts of the genome, right? And the sequencing instruments will give out fragments of the DNA as output. Now, there is no ordering among these fragments. And by default, these fragments are not useful to us unless we develop some computational technique to draw some analysis from this data, right? So the genome assembly problem is to take these individual pieces and reconstruct back the original genome. So think of it as like if you, let's say, imagine you give a dictionary book in the hand of a kid, and the kid just tears the book into pieces. And now you come back and you want to stitch together those pieces back into the book. So that's a very layman description of this problem, that you have small pieces of the string and you want to reconstruct back the original genome from these pieces, right? So what is the technique that we use to solve this problem, right? So think of it as a graph problem where intuitively what you want to do is to analyze overlaps among these fragments. So these fragments will likely have overlaps among them. And those, if the two fragments have overlaps, which means that maybe you want to join one fragment to the second fragment based on the overlap information. So if there's a string overlap between two fragments, maybe you want to join these two fragments to form a single string. So this gives rise to an interesting graph problem where if you think of each fragment as a vertex in the graph, then edge indicates the overlap between two fragments. The genome reconstruction problem becomes the challenge of identifying a suitable walk in this graph that spells the original genome. Right? So maybe the original genome starts from this vertex and you have to compute a walk along this graph that spells the original genome. So this is the applied graph problem where we use various techniques and scalable algorithms to resolve the graph and ultimately give out some genome reconstruction back to a biologist so that they can do further downstream analysis from the genome. So this is one example of the project where we are interested in reconstructing back the genome from small fragment strings using graph algorithms. Okay, so the second project is related to scalable string pattern matching. In the string pattern matching, as I said, when we run a sequencing instrument, we get short pieces from the instrument. Now, one technique that I described previously to reconstruct the genome was using a graph algorithm. But sometimes in some problem categories, we can also use a reference template. So for example, if I'm sequencing a human genome of my relative, maybe there is a human genome already sequenced and known to us where we can map those fragments that I just sequenced to the template and know the differences in the sequenced genome with respect to the reference. So think of it as a string pattern matching problem where I want to pattern match the individual fragments to the reference. So for example, here, this fragment CACAT looks very similar to this region of the reference, CTCAT. So this tells me that there's a mutation at this position of the genome where a character T has been mutated into character A. So one can use a reference template, employ pattern matching algorithms to match these fragments and understand the differences in the sequence genome. So you can know what are the mutations in the sequence genome, and that may explain what are the disease or what are the genetic disorders that are present in the genome, right? But what are the computational challenges here, right? So again, scalability is important because if you are generating terabytes of data from a sequencing instrument, the algorithm has to be really fast. Otherwise, we won't be able to finish in a timely manner. The second is error tolerance. So these sequencing instruments are not perfect. They will make mistakes while reading the DNA. So for example, some of the characters may have errors. This C may be actually A in the original genome. So there is some corruption introduced by the sequencing instrument themselves. So the algorithms, the pattern matching algorithms have to be error tolerant. They have to allow certain level of errors while doing pattern matching. So there are similar domain-specific challenges which we address in this problem and ultimately give out the list of mutations that the sequence genome have based on this analysis. Similarly, so the last example that I want to give in this presentation is related to compression of genomic data. So in this project, we are working as part of a national consortium, and this is a Genome India consortium where 20 institutes across the country are involved in sequencing and analyzing 20,000 human genomes. So these 20,000 humans are coming from different states of the country, from different tribes, different, to cover all the diversity in the Indian population. Now, there are several biological questions that will be analyzed using this project. But on the computational side, also, there are many interesting problems that have been motivated by this project. For example, one problem that we are addressing in our lab is how to compress this data. So each sequencing sample from a single individual consumes 100 gigabytes of space on disk. Now, if you add all of them up, we are talking about 2 petabyte space on disk. So the question that we are addressing in this project is how do we efficiently compress this data to make it use as small space as possible. Now, of course, there are many reasons why we want to do this. First is, of course, to use as much less space, because less space means we have to spend less infrastructure cost in storing this amount of data. The second application you can think of it is if you want to download this data, if you think about download 2 petabytes of data from a server, even if you have access to it, downloading 2 petabytes of data is not possible even on IISc network. If we talk about 3G, 4G networks, we cannot even think about downloading 2 petabytes of data in a reasonably efficient manner. So compressed data is also useful for enabling people to have access to this data across the country. Yeah. So this project concerns about compressing the genomic data, and naturally we want to use the redundancy in this data to compress it as much as possible. So one may ask, why don't you use gzip? So gzip doesn't know about the specific domain characteristics of this data. So the algorithms that we develop are domain specific, which use domain specific properties in this data. For example, one obvious property is that the DNA sequences are only containing of 4 alphabet, like A, T, C, and G. So it's not a general English text. It has a special structure in the text, in the data, which we have to use to develop better compression. Yeah, so this was to give you a flavor of the problems that we work in our lab, right? So again, as you can imagine, because of the interdisciplinary nature, we work with different institutes across the country and outside the country as well. So again, this includes both academic institutions, hospitals, as well as industry, right? So just to, I also want to mention some of the recent lab achievements. So despite that we just started our lab last year, Two students of our lab were part of the winning team in National High Performance Computing Hackathon held last year, right? Software developed by us are widely used in the community. For example, I have listed two examples here. Both of them have more than 10,000 downloads by various users around the globe. We also publish in leading journals. So you are welcome to check some of the recent publications if this work excites you. And yeah, we have research support from various Indian as well as foreign agencies to do our research. So from infrastructure wise, we have access to various high performance computing resources. So all the students in our lab use these high performance computing resources. And in fact, no work is done on local desktop because the data is huge. So we cannot work on local systems. All the work has to be done on remote HPC facilities. Yeah, so just some frequently asked questions for the prospective students. So one frequently asked question is, do I need a background in? So just can you please mute yourself? I'll take the question at the end. Okay, so yeah, so in terms of background, so students often ask whether they need a background in biology or genetics before joining the team. The answer is no, but you will need the curiosity to learn basic biological concepts. In fact, in our lab, we do have to read both computer science papers as well as biology papers. We cannot overlook one side because we are working on in an interdisciplinary area. So we have to understand both the biology side of the problem. Otherwise, we may address a problem which doesn't have any application. So that curiosity is always needed and we have to learn as we go. The prerequisite for joining this lab is that we are looking for students who have a strong background in algorithms and data structures, as well as strong programming skills, preferably in either C, C++, or Java. The reason for these languages in particular is because when we are working with large amount of data, we need to develop software that can scale. And often these are the languages which we have to use to develop scalable software. So finally, I'll close with saying that this is the lab URL. All the information about publications, software, students, their contacts. You're welcome to contact us for any information if you're interested in joining our lab. The interview, in terms of interviews, again, the prerequisites indirectly tell you what topics we will be interviewing in. So we are looking for students who want to dive into interdisciplinary research at the intersection of computer science and biology. And they already have fundamental computer science and programming skills. Okay, so with that, I'll close here and I'm happy to take questions. So let me stop sharing my screen. So feel free to unmute yourself if there are any questions.
So Chirag, we have a couple of like 5 or 10 minutes and I don't see any hands from the students. So maybe you can tell a little bit more about yourself so that the students know you were, what you did and things like that.
So that's a good idea. So I did my BTech in computer science from IIT Delhi. While doing computer science in my final year, I got interested in learning more about biology and applications of computer science in biology. So I decided to pursue PhD in a lab which is also doing computer science and high performance computing research, but with application in molecular biology. So I went ahead and did my PhD in Georgia Tech, the Department of Computation and Science. So it's called Computational and Science Engineering, which is very similar to CDS. So it has a very similar nature of conducting interdisciplinary research as we have it in our department, right? So after I finished my PhD, I did my postdoctoral work in National Institutes of Health in Washington, DC. So that was in, so that's a biomedical research facility. So that again wanted to get more exposure on the bio side of my problems that I was addressing. So yeah, so that's the background I have. So getting exposure to both computer science as well as biomedical facilities during the career. And I started as a faculty in IISc in October 2020. Yeah.
Thank you, Chirag, for the very nice presentation about your lab. And if students, if you have any more questions, feel free to ask them. Professor Chirag is available for five more minutes. So please go ahead if you have any questions.
Hi, sir. I have a question. The brochure mentions that you will be asking graph algorithms as well in the interview.
Yeah, so basically, I mean, if you think about any general algorithms course that is taught in generally taught in UG computer science programs, the graph algorithms is also part of it usually. So that's why I included graph algorithms. And as you saw in our presentation also, the graph algorithms are important for many of our projects. So that's why we asked about graph questions. Yeah.
My question was like, up to what level of graph theory you will be asking?
So you can again you can check as I said UG level. So you have to check the you can check any standard algorithms course taught at any of the institutes, and so generally the standard algorithms, graph algorithms, right? Yeah.
Now, Chirag, I think the question is mostly about. So do people need to know the names of the different algorithms that are there, or what are you expecting them, right? So do they need to think through the problems, or like, so will the questions be more like, okay, go ahead, explain Dijkstra's algorithm? Will it be like that, or will like that? That's what the question is. Yeah.
Right. So yeah. So the questions will check the understanding of those algorithms in the sense that it won't be a direct question like so for example it won't be a direct question like what is a Dijkstra algorithm. It may be a question which may use Dijkstra algorithm, but it won't be told to you. So one has to connect the pieces, but as long as you need know fundamental algorithms like Dijkstra, breadth first search, depth first search, and yeah, so some of the fundamental algorithms that are taught in UG courses have to be known.
So I have one question. Am I audible?
Yes, Vishal.
So as in the third project, you have told about the compression of genomic data, like you are working with the data presented by our country and the people of our country. So basically we have to deal with the demographics and about the people, or we just have to worry about the algorithmic part. We don't have to go for the regional geography or the demographics of a particular place from the data we are dealing with. Like we don't have to work on them, or we also have to, because I am personally interested in that part also, like I'm very much interested in geography and demographics of a place. So that's why I think this is the right lab for me to work, because this is a very good intersection of algorithms and biology and geography, right? So my question is, so working on the genomic data compression, like we have to study about the demographics of a particular region of our country, or only we have to deal with the algorithmic part?
See, so I mean, the end goal of the project, the output that we want ultimately is the compression efficiency of that project. So in that project, what we are interested in is to compress the data in as small space as possible. Now, as part of the algorithm, maybe you can use the demographic data to improve your algorithm. For example, if you find that, okay, clustering those genomes which are coming from a single state or single tribe, if there is a compression algorithm that makes use of the demographic data also in improving the compression efficiency, that would be interesting. So
Yes,
far,
sir.
we have not looked at the demographic information in our algorithms.
No, sir.
But definitely that, yeah.
My point was basically we should be knowing which data we are dealing with, right, sir? That's what my point was. See, basically we have to study about the demographics of the data that we are dealing with, or just the algorithmic part that we have to concerned about.
Yeah, so so the goal of the project is to develop an algorithm that targets best compression. So in the end, our goal is on the algorithmic side.
Okay, sir.
Demographics is handled by the actual field workers. So they will decide, okay, which tribe
Okay,
to
sir.
target, uh, which tribe to target and how many genomes to sequence from which state and so on. Yeah, so but the data we don't have any control of what data is coming to us.
Okay, sir. Thank you very much.
So we'll take one last question. Abhishek, go ahead, Abhishek.
Am I
Abhishek.
audible, sir?
Yes, go
Yes.
ahead.
I would like to ask, sir, since I'm from electronics background, sir, I'm not very much conversant with this algorithm, sir. But I'm really keen into getting into ATCG department of CDS. Sir, how suppose what should be my approach for the interview preparation?
Okay, so my suggestion would be to still develop. So there are lot of material on algorithms and data structures. So if you tell us, okay, even if you have not taken the full course, if you go and prepare some specific part of the course, we want to know your learning ability. Like if we can comprehend your learning ability in that topic, and we see that, okay, once you join IISc, you can take the course and learn the remaining topics easily. That is good enough for us, right? So if you tell us, okay, this is the part in the algorithms topic that I came prepared on, we can only test you on that topic. So it's okay to not have undergraduate degree in computer science, and because all of the students who join our lab, most of them do take algorithms course after joining the lab. So irrespective of whether you have CS background or not, you can always acquire the necessary knowledge.
Thank you, sir.
Okay.
Yeah, thank you, Chirag. Thanks for the very nice presentation about your lab, and hope the students have clarified their questions as well. So thank you. We'll now move on to Professor Ratikanta Behra. Can you please share